All right, welcome to the final video for Chapter 15. In this video, we're going to look at the details of translation. How do we get from messenger RNA to our polypeptide or protein product? Interestingly, the synthesis of proteins takes up more of a cell's energy than any other metabolic reaction that happens in the cell. Also, this kind of goes hand in hand. Proteins actually account for more mass than any other macromolecule in our living organisms, in our cells. Proteins are extremely important in terms of structure and function. They act as enzymes to catalyze reactions, and they're produced through the process of translation. So translation allows us to produce proteins, and it occurs when we decode the sequences in messenger RNA, and we saw those codons earlier, in order to add amino acids, link them together to form a polypeptide chain, and that will ultimately create a three-dimensional shape or fold into a three-dimensional shape to generate our final protein product. So these polypeptides are made of subunits or monomers of amino acids. And amino acids, when they're linked together, they are connected through a type of covalent bond known as the peptide bond, where we're going to see the amino group of one amino acid binds to the carboxyl group of the next amino acid. Ribosomes will catalyze this process of protein synthesis, and we're going to see ribosomes are composed of both proteins and ribosomal RNA. Protein sizes vary dramatically. Some of them are very small, consisting of about 50 amino acids, and others are very large, and they can be made up of more than 1,000 amino acids. We've seen an image like this before, way back from our first exam material. Here I have one generic amino acid on the left, and just as a refresher, remember we always have a central carbon, an amino group, a carboxyl group, a hydrogen, and then the R group or side chain. And that's really the difference between the 20 amino acids. Here we have another amino acid, and the peptide bond will be formed between the carboxyl group of one amino acid and the amino group of the next amino acid. So I can see that water will come out of the reaction. This is a dehydration synthesis reaction, or sometimes it's called a condensation reaction. So here I have my peptide bond. The ribosome is the main structure that's responsible for translation. It's going to be reading the messenger RNA and translating that into protein. If I look at the structure of the ribosome, it's made of a small subunit and a large subunit. Both subunits will have, in combination, so if you look at the entire ribosome, it's composed of polypeptides and ribosomal RNAs. And some of these ribosomal RNAs will have a catalytic function. These ribosomes differ depending if you're looking at prokaryotes versus eukaryotes. In prokaryotes, the small subunit is the 30S subunit, the large subunit is 50S, and in combination, the entire ribosome is 70S. And this is not additive. The S refers to a Svedberg constant, which you'll learn more about in chemistry, but it basically has to do with sedimentation and how far down a centrifuge tube these subunits and the ribosome will travel down when you spin it down within a centrifuge. In eukaryotes, they're larger. This is the small subunit, 40S, large subunit, and combined we have an 80S complex. The nice thing about the difference between prokaryotic and eukaryotic ribosomes is that they're made of different amino acid sequences, and there are some antibiotics that take advantage of this difference, where we have antibiotics that can prevent or block prokaryotic ribosomes from translating something without blocking eukaryotic ribosomes because of that nice difference. If I look in terms of their function, the small subunit will be bound to the messenger RNA. You can see that over here. The large subunit will be binding to transfer RNAs and allowing that polypeptide to form. Continuing with the ribosome structure, if I have the entire ribosome, small and large subunit together, I can see that there are 3 transfer RNA binding sites. And from left to right, they are E, P, and A. E is the exit site where transfer RNAs will leave. P is the peptide or peptidyl site where we're going to form peptide bonds between the amino acids. And A is the aminoacyl site where new transfer RNAs are going to enter, bringing in the amino acids that correspond to the codon found on the messenger RNA. So I have those listed here. A site is where the new tRNA enters. P is where peptide bonds will be formed, and this will be catalyzed by ribosomal RNA found in the large subunit. And E is where the discharged transfer RNA will leave to get a new amino acid attached to it. So you can see that these transfer RNAs have an amino acid attached, but by the end of the process, they no longer have an amino acid attached, and those are known as discharged transfer RNAs. They need to get recharged. They need another amino acid to be placed at this end. So during translation, we're going to see that charged, the ones that have amino acids attached to the tRNA, charged tRNAs are going to enter the A site. The acceptor site is sometimes what it's called. And the anticodon of the transfer RNA is going to be complementary to the codon on the messenger RNA. And the codon on the messenger RNA determines which amino acid is attached to the tRNA. So here I see, I think it's GGG, so that must encode glycine for the amino acid. Here's a closer look at that bottom picture. Here is my messenger RNA, and we're going to see that translation occurs with the ribosome starting at the 5' end of messenger RNA and moving down towards the 3' end of the messenger RNA. So after we have our charged tRNA entering the A site, the next step is that a peptide bond will form between the amino acids. So it's kind of weird, it's not shown in here, but what's going to happen is this is going to be removed from the P site, and all of these amino acids will form a peptide bond with this new amino acid. This whole thing will shift over here, and then this will be discharged. It will no longer have anything attached at this end, and this is because this whole ribosome complex will be moving forward by 3 nucleotides, by another codon. So everything shifts forward. This one will become discharged, and everything, this one will be shifted over here. And then there'll be space for the next transfer RNA to come in through the A site again. So how do our tRNAs, our transfer RNAs, get charged with an amino acid in the first place? This is done by aminoacyl tRNA or transfer RNA synthetases. So these are the enzymes that charge each transfer RNA with its correct amino acid. And this is based on the anticodon sequence on the tRNA, or you can say it's also based on the codon sequence that it'll be complementary to on the messenger RNA. So these are complementary to one another. There's at least one aminoacyl transfer RNA synthetase for each amino acid. So they're unique to which amino acids they're going to charge or add to each of these transfer RNAs. This process does require energy in the form of ATP. What happens is ATP will be hydrolyzed or broken down into adenosine monophosphate, and you'll see a pyrophosphate molecules released. The AMP, this adenosine monophosphate, will be temporarily bound to the amino acid. And then when the amino acid is combined or charged to the transfer RNA, the AMP will be released. So similar to the problem I had you guys practice at the end of the previous video, if I look at which amino acid belongs to the end of this transfer RNA, I just look at the anticodon, and I can use this chart. I know if this is the anticodon, then the codon on the mRNA, if this is the codon, would be complementary, so it would be U, G, U, and I'm looking for the sequence. UGU, and I see cysteine. So that is why cysteine is the amino acid that will be attached to this transfer RNA. Just as in transcription, for translation, we also have three basic steps: initiation, elongation, and termination. And we're going to go through each of these steps in detail in the next several slides. We're mainly going to focus on translation in prokaryotes because this is more well studied, more well known. Let's start with initiation. So this is initiation. What happens is the initiation complex will form. The messenger RNA is located here, and what we're going to see is the first thing that happens is the small subunit of the ribosome will bind to the messenger RNA. How does it know where to bind? It's going to look for the start codon, AUG, and it's going to center that in the P site. Remember, the sites were E, P, and A. So the P site is where AUG will be found. And there we're going to have a special transfer RNA known as the initiator transfer RNA that will bind to that codon. So the anticodon of that tRNA will be bound to, or base pair, with the codon. Remember that AUG encodes methionine, and if it's in prokaryotes, we said it was formal methionine. So this is in prokaryotes. And then, now that we have everything down, then the large subunit of the ribosome can come in and cap everything off. So if I look at differences between prokaryotes and eukaryotes, for initiation in prokaryotes, the ribosomal RNA in the ribosome will recognize a specific sequence right before the start codon, and that's something known as the Shine-Dalgarno sequence. So that will be upstream to the left of, in this picture, of the AUG site. So this is for prokaryotes. In eukaryotes, the initiation complex recognizes that 5' G cap on the messenger RNA, and that's where it binds and looks for the first AUG, or first start codon. The next step is elongation, and during elongation, the transfer RNAs will move in just as we saw in the previous picture. They're going to enter the A site, and these will be charged tRNAs that carry the appropriate amino acid depending on the anticodon or codon. Then what we're going to see is that peptide bonds will be formed, and this occurs through an enzyme known as peptidyl transferase, and this will be found in the large ribosomal subunit in the form of a ribosomal RNA molecule. So this is interesting because it's an enzyme that happens to be an RNA molecule. This will allow peptide bonds to be formed between the new amino acid that just came in and the growing chain. And then again, everything is going to continue shifting 3 nucleotides to the right to read the next codon on the mRNA sequence. In prokaryotes, this is super fast. So for prokaryotes, they can translate about, let me write this down. For prokaryotes like bacteria or like E. coli, they can translate about 200 amino acids in about 10 seconds. So much more rapid than eukaryotes. Finally, in our last step, termination, what happens is in the A site, we're going to reach one of those 3 stop codons we learned about earlier. If you see any of these in the A site, protein release factors will come in and bind the A site because there's no amino acids corresponding to these stop codons, and then everything will fall apart, and the amino acid chain or that polypeptide will undergo further processing, especially if we're looking at eukaryotes. This usually means that the amino acid chain is going to move into the endoplasmic reticulum, usually the rough ER, to be processed further. So here's a closer look at that termination process. Again, in the A site, we're going to reach one of those 3 stop codons. Since there's no transfer RNA corresponding to any of these stop codons, protein release factors will bind to that A site, and it's going to catalyze the hydrolysis between the last amino acid and its transfer RNA. This is going to cause the release of the polypeptide, and then GTP will be used in order to disassemble the ribosomal subunits. The large subunit and the small subunit will break apart, releasing the messenger RNA as well. And if we look at eukaryotes, I don't have it on the slide, if we have eukaryotes and you look closely at that polypeptide sequence or amino acid sequence, at the amino end of that protein, there is going to be a certain sequence known as the signal sequence. And the signal sequence directs the polypeptide as to where to go next for processing. This will allow the polypeptide to move to the right compartment within the cell at its next step. And our book calls it the train ticket to its next destination. Once it reaches that destination, the signal sequence will be removed. And you can see that in this picture, the signal sequence is removed when we reach the ref ER. For proteins, once they're completed, once they've reached their destination, they will fold. So they need to fold into their three-dimensional shape. Sometimes this occurs spontaneously just because of the side chains or R groups that are found in the amino acids. Sometimes it requires help, and this is completed through proteins known as chaperone molecules. Remember from before, we talked about proteins and their three-dimensional shape are sensitive to the environment. So this requires an optimal or normal temperature for that protein, as well as a normal or optimal pH that's specific to that protein as well. And that takes us to the end of chapter 15. In the next chapter, chapter 16, we're going to be looking at gene expression. How do we determine or control when our genes are transcribed and translated?